In this section, we're going to talk about how GloCyte is innovating CSF cell counting. Advanced Instruments has a suite of state-of-the-art tools designed for the optimization of laboratory testing, culture growth, and even pipette calibration. The GloCyte automated cell counter for CSF is designed to help ease the staffing burden and requirement for advanced technical expertise by reducing the time and workload endured by medical laboratory staff to regularly prepare and interpret manual cell counts. GloCyte was awarded FDA clearance to provide the quantitative determination of fluorescence-labeled total nucleated cells and erythrocytes in cerebrospinal fluid collected from adult and pediatric patients in 2016, following the successful completion of clinical trials at 5 highly renowned healthcare institutions. 2 peer-reviewed publications were published by the clinical trial principal investigators following the trial. One in the International Journal of Laboratory Hematology, and the second in the American Journal of Clinical Pathology. The GloCyte automated cell counter system is intended for use by trained healthcare professionals in clinical laboratory settings to provide quantitative determination of fluorescence-labeled total nucleated cells and erythrocytes in cerebrospinal fluid collected from both adult and pediatric patients. The GloCyte test works by using 2 reagents, one to stain the TNCs and a second to bind a fluorochrome-labeled antibody to the surface of the RBCs in the sample. These samples are then pipetted into the test cartridges. The CSF is pulled through the membrane when a vacuum is applied, and the cells present in the sample are then captured on that membrane. Images of the cells are captured with a digital CCD camera and fluorescent microscopy, and then each cell type is counted via a sophisticated algorithm. The steps below will be outlined in a video on the next slide. The GloCyte test principle enables the automation of CSF cell counts down to 0 cells per microliter, outperforming the low-end limitations of other automated cell counters designed for the enumeration of cells and body fluid. Additionally, GloCyte performs well within the precision acceptance criteria for 20% for both TNC and RBC counts tested across a range of standards. Minimal variation in cell enumeration was reported between runs, days of operation, sites of operation, and operator, all falling well within the acceptance criteria. Method comparison results also met acceptance criteria and demonstrated that GloCyte yields results equivalent to manual counts using the Neubauer hemocytometer for both pediatric and adult populations with slope, intercept, and biases all well within the acceptable range for all applications tested. Acceptance of the slope criteria is further demonstrated on these passing Bablock plots for each application tested. As previously mentioned, GloCyte can detect cells down to 0 cells per microliter, which means that the limit of blank for both TNCs and RBCs is less than 1. The limit of detection for both TNCs and RBCs is 1, and the limit of quantitation for both TNCs and RBCs is 3 and 2, respectively. All of that testing leads to this, the exceptional reportable range of the GloCyte analyzer that allows us to report down to 3 cells per microliter of TNC and 2 cells per microliter of RBC, offering physicians with the ability to determine if the TNC count lies within the normal range for CSF or if it is slightly elevated into the abnormal count of greater than 5 cells per microliter. As far as assay interference goes, again, GloCyte falls well within the acceptance criteria with one important caveat to consider for you and your customers. TNC reagent stains all nucleated cells, so nucleated red blood cells will not only be stained by the RBC reagent, but also by the TNC reagent as well.